we will further consider the various subtypes of hemolytic anemias. Now, hemolytic anemias is quite a large topic. There are various causes of hemolysis, and again, to make things easier, we are grouping them into various categories. First, we consider genetic defects. The genetic defects can be either inherited or it can be acquired. The second major group would be immune mediated hemolytic anemias. The third one would be anemias resulting due to mechanical trauma. The fourth one is hemolysis occurring due to infections. The fifth one is hemolysis occurring due to toxins. The sixth one can be hemolysis occurring due to sequestration in the spleen or hypersplenism. The first subcategory of this, that is genetic defects, under the inherited group, again you have various types. Firstly, we will consider anemia due to an inherited genetic defect affecting the red blood cell membrane. Now, the red blood cell has, again, we've always discussed this about the shape of the RBC. It has a beautiful dumbbell shape, and this shape is maintained by the cytoskeleton. There are various proteins which are important in the maintenance of this shape of the RBC. These proteins include spectrin, ankyrin, band 3, band 4.2, etc. Defects in these proteins can sometimes lead to a weakening of the red cell membrane. So, for example, the commonest defect leading to this particular condition of hemolysis due to membrane abnormality is a defect in ankyrin. When ankyrin is lost, when the protein ankyrin is absent, what happens is small amounts of the red cell membrane are lost, and ultimately this red cell, which had a dumbbell shape, it is forced to take a shape with the least surface area because of the loss of membrane. This shape with the least surface area is a sphere. So, this is the reason for hereditary spherocytosis, a defect where there is inherited genetic abnormalities in the cytoskeletal proteins of the red blood cell. Apart from ankyrin, band 3, band 4.2 and spectrin are also sometimes mutated in hereditary spherocytosis. How does spherocytosis lead to hemolysis? Now, remember, if the red blood cell has to go through the spleen, it should be flexible. It should have a thin waist. When it loses membrane, it is turned into a round cell. A round cell is not flexible. So, a round cell gets entrapped in the spleen. It leads to extravascular hemolysis in the spleen. Again, since the spleen is the major site of hemolysis here, a splenectomy is helpful in many of these patients. Now, we will consider another hemolysis which occurs due to an inherited genetic defect, but it is not in the cytoskeletal protein, but it is rather in a RBC enzyme. This particular enzyme is called glucose 6-phosphate dehydrogenase, and the function of this enzyme is it helps in the conversion of glucose 6-phosphate to 6-phosphogluconate. In this reaction, one molecule of NADP is reduced to NADPH. This reduced NADPH is important in converting oxidized glutathione to reduced glutathione, and this reduced glutathione helps in the function of an enzyme called glutathione peroxidase. Glutathione peroxidase is an antioxidant. This particular enzyme helps in the conversion of hydrogen peroxide to harmless water and oxygen. So, hydrogen peroxide, which is a reactive oxygen species, it is a free radical derived from oxygen. It is it can cause oxidant damage to cells. These cells are protected from this oxidant damage by the presence of or the proper activity of glutathione peroxidase. If glutathione peroxidase has to act, it needs reduced glutathione. For reduced glutathione to be produced, it needs reduced NADP, it needs NADPH, and NADPH is produced by the action of glucose 6-phosphate dehydrogenase. So, if the person is deficient in glucose 6-phosphate dehydrogenase, he tends to develop oxidant damage to his RBCs. This kind of damage usually occurs when there is exposure to oxidants, like for example, when there are intake of drugs like antimalarials, or when you take food like fava beans. All this can lead to damage of red blood cells. We have now considered two forms of hemolytic anemia arising due to a genetic defect which is inherited. Now, the next type of inherited genetic defect which can lead to hemolytic anemias affect the hemoglobin molecule as such, specifically the globin chain. The defect in the globin chain can be either qualitative or it can be quantitative. This defect, if it is qualitative, you have an abnormality which is called sickle cell anemia. The defect here is in the beta chain of hemoglobin, where there is a mutation, a point mutation, which leads to the substitution of glutamate for valine in position 6 of the beta chain. This genetic defect leads to an abnormal hemoglobin. There are two types of patients who can carry this hemoglobin. One is where almost the entire hemoglobin is hemoglobin S, or this sickle hemoglobin, and the other one is where only a part of the hemoglobin is hemoglobin S. The first subgroup of patients, they are called or they are said to have sickle cell disease, and the second one is said to have the sickle cell trait. Those who have sickle cell trait do not suffer from massive hemolysis, but those who have sickle cell disease are at risk for hemolysis. Now we will see why by just looking at the pathogenesis of this mechanism, the mechanism of this hemolysis. What happens here is the sickle cell, the sickled hemoglobin, it has a tendency to polymerize whenever the oxygen tension comes down. So when there is a small amount of deoxygenation, when there is not enough oxygen, the HbS molecules tend to polymerize. So as they polymerize, they form long sticks. Now the initial stage of deoxygenation, this change is seen as a more viscous or jelly like cytoplasm of the RBC, but as it progresses, the RBC is, you could say that the RBC is carrying a long stick of HbS inside it. Now this elongating stick will pull both sides of the RBCs and this ultimately leads to an RBC with pointed ends because there is a stick inside it, and as it further polymerizes, because its length further increases, it curves and forms the typical sickle shape. Now one point to remember when assessing the peripheral smear of such patients is that you do not really have to see a curved RBC to call it a sickle cell. It just has to be an elongated RBC with pointed ends. It does not have to start curving, but because the curve looks nice, being morphologists, we pathologists try to compare it with something that we know, and that is why we call it a sickle cell. Now sickle cells, the problem with sickle cells are they are at a disadvantage when going through the spleen. Now I earlier mentioned that the spleen is like a quality control mechanism, it is like an obstacle course for RBCs. Now just consider how unfair it would be if you were to send a person through an obstacle course with a long stick in his hand. He would definitely not be able to complete the course, he would be trapped inside it. That is the same thing that happens to a sickled hemoglobin, and it gets trapped in the spleen and it leads to extravascular hemolysis. Another complication of such patients is that they tend to have develop microvascular occlusions in areas where there is a little stagnant blood flow. However, since we are considering only anemia today, we are not going into the pathogenesis of that particular mechanism. Now, sickle cell anemia, I said those who have disease have hemolysis, while those who have trait do not have too much of a problem with hemolysis. The reason is those who have sickle cell trait have both a combination of HbS molecules and HbA molecules. Now HbA interferes with the polymerization of HbS. If there is only HbS, it polymerizes. If there is HbA, it comes in between and breaks down the polymer. Therefore, there is no sickling in sickle cell trait. Okay. The next anemia due to an inherited defect in the globin part of hemoglobin is a quantitative defect of the globin. There is a suppression of the globin chains, and this is called thalassemia. Thalassemia refers to the Mediterranean areas. It was first described there, or it is prevalent there. That is the reason it is called thalassemia, and the basic problem here is there is a reduction in the globin chains. Now it can be either a reduction of the alpha chains, which is called the alpha thalassemia, or beta chains, which is called beta thalassemia. Beta thalassemia is slightly more important than alpha thalassemia because it can cause more severe hemolysis. Now I will first describe the cause of hemolysis or the mechanism of hemolysis, and then tell you why beta thalassemia is probably more severe than alpha thalassemia. In thalassemia, what happens is globin chain production is reduced. Now globin chain production is reduced. I earlier mentioned that in the etiological classification of anemia, we have three subtypes: increased blood loss, increased destruction of RBCs and decreased production of RBCs. So, logically, if there is a decreased production of globin chains, this anemia should ideally come under decreased production or the subtype of decreased production anemias, but it is taken under hemolytic anemias because apart from when this globin chain production is decreased, what happens is, say we will take the case of beta thalassemia, there are not enough beta chains, which leads to an accumulation of unpaired alpha chains in the cytoplasm of the RBCs. These unpaired alpha chains are toxic, especially to the membrane of the RBCs. They cause damage to the membrane of the RBCs within the bone marrow. Within the bone marrow, these damaged RBCs are they are channeled into apoptosis. There occurs cell death due to apoptosis. Almost 70 to 85% of RBCs are killed within the bone marrow through the mechanism of apoptosis, and this is called ineffective erythropoiesis. Therefore, even though it is the initial mechanism is a reduced production of globin chains, ultimately it leads to hemolysis within the bone marrow. A small subgroup of red blood cells which can escape from the bone marrow are still have to deal with the spleen because they are not of good quality. Their membrane is damaged. So, when they reach the spleen, the quality control manager in the spleen destroys it, leading to extravascular hemolysis too. So, you have both ineffective erythropoiesis in the bone marrow and extravascular hemolysis within the spleen. I mentioned that alpha thalassemia is not as severe as beta thalassemia. Why is this? Beta thalassemia, as I said, it leads to a decrease in beta chains, therefore there is excess alpha chains, and these excess alpha chains damage the erythrocyte membrane, leading to ineffective erythropoiesis and extravascular hemolysis. However, unpaired beta chains are not as toxic as unpaired alpha chains. So, in alpha thalassemia, you have unpaired beta chains. In beta thalassemia, you have unpaired alpha chains. Unpaired alpha chains are much more toxic than unpaired beta chains. This is why beta thalassemia is more severe than alpha thalassemia. So, this is regarding a few inherited genetic defects leading to a hemolysis. Now, there is a very special type of hemolysis occurring due to an acquired genetic defect. This is called paroxysmal nocturnal hemoglobinuria. It is the only acquired genetic cause of hemolysis. The gene involved is called phosphatidyl inositol glycan complementation group A. If you don't want to remember this big name, you can just think of it as PIGA or PIGA. It is a gene which is responsible for producing a certain amount of molecules on the membrane of cells. Now, if you remember the cell membrane structure, you may probably remember that there are two ways in which proteins are attached to the membranes. The most common type is the transmembrane protein, and a certain subgroup which is rarer is a membrane which is attached through an anchoring molecule called glycosyl phosphatidyl inositol or GPI. This PIGA gene is responsible for the production of GPI, and when PIGA is mutated, there are not enough GPI anchors, and therefore there is a loss of these proteins which are anchored to the membranes through this GPI molecule. So, when there is no GPI, there is no GPI linked proteins, and three important proteins which are responsible for this particular condition called paroxysmal nocturnal hemoglobinuria are CD55, otherwise called the decay accelerating factor, CD59 or the membrane inhibitor of reactive lysis, and C8. Now, CD59 is the most important of these three proteins, and when this protein is absent in the cell, what happens is there is a hemolysis occurring due to complement mediated lysis. So, hemolysis here occurs due to complement mediated lysis. In a normal individual, this complement mediated lysis is avoided by the presence of CD55, CD59, etc. But in those with the mutation of GPI linked of the GPI anchor, they do not have this protection against the complement mediated lysis. Therefore, it leads to hemolysis. Now, we will take the case, in this particular case of paroxysmal nocturnal hemoglobinuria, the affected cell is a stem cell. Therefore, it is not only the RBCs which are affected, but also the WBCs and platelets are suppressed in this condition. The next subgroup of hemolytic anemias is the anemia caused through immune mechanisms. There are again various conditions which lead to hemolytic anemias through immune mediated damage. The usually described example is what is called autoimmune hemolytic anemia. Now, even though this is the more commonly used term, some authors prefer to use the term immunohemolytic anemia because not all cases may be autoimmune. Some cases may be produced through drugs. Therefore, it's called immunohemolytic anemia, and in this condition, the mechanism is very straightforward. There are antibodies against red blood cells. So therefore, depending on the temperature at which the antibodies act, you have both warm antibody hemolytic anemias and cold antibody hemolytic anemias. In these immunohemolytic anemias, or especially in the warm antibody type, what happens is there is a partial phagocytosis of the red cells, just like we see in hereditary spherocytosis. So a part of the cell membrane is lost, and therefore what do you see in the smear? In this condition too, you can see spherocytes because the cell tries to take the shape with the least surface area because of the loss of membrane. So both in hereditary spherocytosis and in autoimmune hemolytic anemia, you get spherocytes, but in the case of autoimmune hemolytic anemia, you can detect the antibodies using the Coombs test. So when the Coombs test is positive, you know there are antibodies active there, and that is a case of autoimmune or immunohemolytic anemia. Another example of the immune system causing hemolysis is when you can have the unfortunate scenario where there is mismatched blood transfusion. If the recipient's blood group is not the same as the donor's blood group, the recipient will have antibody, especially in the case of the ABO system. If the recipient will have antibodies which will destroy the donor cells and lead to hemolysis. Another similar picture of hemolysis can occur in newborns, and that is called the Rh incompatibility disease. And here basically what happens is the mother is Rh negative, the child is Rh positive, and if the mother has been exposed to Rh positive blood earlier, the mother develops anti-Rh antibodies, and these anti-Rh antibodies, being IgG antibodies, they pass through the placenta and cause hemolysis within the child. So that is also an example of immune mediated hemolysis.